afternoon, everyone. Hello? Anybody there? You can now start typing at our chat box. Hello, then Cyril. Ayan. Can you hear me okay? Hello din kay John Paul. Good afternoon. Hey! Thank you, Aubrey, for coming in again. Salamat. Thank you that you can hear me okay. Thank you. Hello to Esther Dawn. Hello to James. Hello to Mary Ann. Hello to Joyce. To Maria Luisa. Hello to Charmy. Hello to Anna Joy. Yan. Let's just wait for others, okay? For the meantime, while we are waiting for them, you may type and chat greetings in our chat box. Kung meron kayong mga gustong batiin, I can read that for you. Hello to Carla Jane. I'm so excited for this afternoon. So, our lecture is still actually in our module 1, and I chose this topic. And we can have some fun activities at the end. So, make sure you stay tuned. Hello to Ray Melvin, to Kim Bernadette. Good afternoon sa inyong lahat. Hello to Jose. You can still type our greetings. Anyone you would like to greet as well. The weather is nice today. It's a bit sunny. Aubrey, where are you watching now? Are you in MSU ba? Please remind your classmates that our lecture class is about to start. We will start at exactly 1:25. Okay, 1:25 I will start the lecture. It will not be long. I only have a few slides, but very important for your activities, especially growing of bacteria. Come on. Hello to Han Rafi. Hello to Aryan. Do we have participants from outside Palawan? Can you please chat that? Hello to Nor Aiza. Hello din to Jhonamie. Come on! tell others we are about to start our lecture. So let's just wait for them. Just a little bit more. Maybe some are having troubles with internet connection. So let's give them a bit of time. Ang siseryoso naman, walang mga pa-shout out, walang mga pabati. Oh. Is everybody okay? Walang mga may sakit ba dyan? Sana naman wala, iingat tayo parati. Hello to Willie May. Good afternoon din at sa inyong lahat. So, saan na kaya yung iba? Where are the others? My neck itches. Hello to Florida Lisa. I'm supposed to have around 50 students in 2 sections of microbiology, plus friends from different places, from Palawan, from Mindanao, and from the different parts of the Philippines. From Ray Melvin, pa-shout out po sa module kong Natenga at pa-shout out po sa Galicia family sa Tinigiban. Nako, bakit naman Natenga ang module? Nasabi mo na, but sige lang Ray Melvin, habol ha? So, shout out to Galicia family. Sayang, dapat napanood nila ang pa-shoutout mo, Ray Melvin. We are going to start our lecture in 2 minutes. Hello to Jhonamie. Get ready. We are going to start our lecture in 1 minute. So you listen carefully, you take down notes. Note of the important points, because those may come out in our quiz B this afternoon. Not quiz B, but anyway, activity this afternoon. And who knows, you might win a prize. Okay, from Jose Bernardo Jr. Ma'am, pa-shout out po sa mga classmates kong busy. Second year BS Ed Science 2A. Ayan, sana in-invite mo silang mag-attend lahat. Para marami. Busy sila, ikaw hindi ka busy. It's gonna be an advanced knowledge for you. Okay, so it's 1:25. We're going to start our lecture, the basics of bacteria. So let's just make them catch up. No, anyway, anytime naman na pumasok sila, they will be able to listen to our lecture. And for the parts that you missed, pwede namang balikan later. Kasi automatically, this lecture will be recorded and will be posted in our YouTube channel. So, YouTube channel natin yan. Kasi that is our platform by which we can live asynchronous learning. So, our lectures are all just there. Hello, good afternoon din sa kay Jess. Okay, so I'm gonna start now. So, the basics of bacteria. Now, when we hear the word bacteria, what is it that first comes into your mind? Yeah? So probably, you know, when you talk with your friends, when you talk with your family members, and then you mention the word bacteria, what is it that, what reaction do you often get? Or you yourself, ano ba ang reaction mo pag narinig mo yung word na bacteria? Now, sometimes we think of the words germs, microbes, ano pa ba yan? Diseases, yucky, kadiri, dirty. Diba ganon? That's what we usually associate when we hear the word bacteria. O from Rafi, sabi niya, dirty. So, ayan, no? But, that is putting out of balance. Kawawa naman yung bacteria. Kapag narinig natin ng bacteria, madumi agad, katakot siya agad. But that's not really the case. Although, bacteria are the oldest and the structurally simplest and most abundant life forms. Pinakamarami sa lahat ng life forms. And the groups of which are the Archaea Bacteria, the Cyanobacteria, the Eukaryotic. Abe, sige nga, can you guess? Kung meron bang 100 na bacteria, ilan kaya doon sa 100 na yon ang nakakakos ng disease? Sige nga, type nga daw kayo. Hula, ilan sa 100 na bacteria ang nakakakos ng disease? Or in other words, we call them pathogenic. How many do you think out of 100 bacteria would be pathogenic? Thinking that they are the most abundant. That means they are also cosmopolitan. They are ubiquitous. Ano ibig sabihin nun? We see them everywhere and almost in all types of environments. Pero pag sinabi natin na pathogenic, nakakakos ng disease. So sabi ni John Paul, 10 out of 100. Not bad. Sabi naman ni Rafi, 80 out of 100. Sabi ni Densreal, 1 out of 100 with a question mark. Sabi ni John Paul, 10%. O, pareho lang yan, 10% tsaka 10 out of 100. Oh. So, yung sagot actually ni John Paul at tsaka ni Densreal na may question mark, hindi masyadong nalalayo, no, sa katotohanan. Sa katotohanan. So, tiningnan ko kung may mga bago ba kanina update. So, sabi ni Google, around 5% ng bacterial species daw are pathogenic. Yan, no, out of 100, it means mga 5, no, 5. So, pero, usually, bacteria are associated with diseases and death. So, kaya kinatatakutan talaga natin. That's why, disproportionately, na naiisip natin na karamihan talaga sa kanila pathogenic. Kagaya lang, halimbawa, ng sinabi ni Rafi, 80 out of 100. O kahit naman ako, kung mag-iisip at hindi ko talaga, wala pa akong background sa bacteria, iisipin ko na ganun kadami or mas marami pa, baka sabihin ko 90 out of 100 ang pathogenic, no? So, yes, that's a close call. Yeah. And sabi naman ng isang published paper, the corresponding percent of potential pathogen daw na bacteria, ito siya sa urban setting, is around 3.78 to 4.08%. So, ang baba-baba lang, 'di ba? Out of 100, sabihin na lang natin, 3 to 5 pathogenic microorganisms or pathogenic bacteria ang meron. Sa handouts ninyo, sa inyong mga module ang nilagay ko dyan, there are 87 beneficial, 10% opportunist, and 3% pathogenic. All in all, parang kapag ka 10% opportunist, may possibility din siya na maging pathogenic. So parang all in all, nasa 13% ang pwedeng maging pathogenic. But that is still way smaller compared to 87% na beneficial na pwede nating makuha out of the bacteria. Ano-ano ba mga beneficial effects, beneficial things that these bacteria are giving us? No, 'di ba madami? Oh. Ano-ano 'yun? We use them for bioremediation, pang clean up, pang solve ng mga problema, kagaya ng mga oil pollution, heavy metal pollution, etc. Yung mga kinakain natin, marami doon ang bacterial products. Yung suka pa lang kaagad, bacterial product yan. Yung mga Yakult natin, yung mga yogurt natin, bacterial products yan. Kagaya lang din halimbawa ng mga favorite natin na fermented food, kagaya sa akin, favorite ko ang kimchi. Bacterial product din yan. Those are actually what we call the probiotics. No, yan. Importante na magkaroon tayo ng probiotics para okay ka tiyan. Diba? Okay yung tiyan natin. So, first group of bacteria is what we call the Archaea Bacteria. These are the microorganisms or the bacteria which are confined to the extreme environment. Meaning, ito yung mga environment na yung ibang microorganisms, ibang bacteria, hindi na magsusurvive, but they survive. Kagaya lang halimbawa na mga hot springs na mayroong volcano, yung deep sea vents, yung deep sea vents, yan yung parang may volcano din na bumubuga sa ilalim ng dagat, no? Ganon. O kaya very cold places like in polar regions, where there is iceberg, kagaya ng kung nasaan nalubog ang Titanic. Alam mo ba na nung tinignan nila, binalikan nila uli kung saan nalubog ang Titanic, nakakita sila doon ng mga metal-eating microorganisms, metal-eating bacteria. O, diba? So, these bacteria are what we call the Archaea Bacteria. Sila yung mga extreme, kaya nilang mabuhay sa mga extreme na cases. Kagaya lang ng methanogens. Itong methanogens, they usually exist and survive in areas which are anoxic. Walang ibang source ng air or oxygen, for example. Kagaya lang ng sa mga sewage. So, ginagamit siya for sewage treatment, for methane gas production. So, marami ito dyan sa mga septic tanks. For marshes, for swamps, including sa gut ng animal, kagaya ng cow. Ano bang nagkukos kung bakit bumubuga ng maraming methane ang baka when it farts? Diba? That's because of the methanogens living in the gut of the cattle. Ayan. And then, next, we have halophiles. Pag narinig nyo na yung word na halophiles, halo means, has something to do with salt. Phile, from the word philic, halophilic means loving, salt-loving. Example, the Great Salt Lake and the Dead Sea, napakataas ng salinity, higher than 300 parts per thousand. Ay, yung dagat natin dito, naaalatan na tayo, but that is only around 30 to 35 parts per thousand na salinity. Ay, paano naman sa Dead Sea? Kung meron ka mga 300 plus ppt, o di, meron pa kayang microorganism na magsusurvive or other organism, halos wala. But bacteria can, because some of them are called halophiles, halophilic. They love salt. So, because they have the enzyme active only at high salinity. So, halophile ang tawag natin sa kanila. And then another group we call the Thermo Acetophile. So, ito naman a combination of high acidity, ibig sabihin very low ang pH, at saka very high temperature. So, they can stay in 60 to 80 degrees centigrade. Ay, luto na yung itlog dyan. Diba? 60 to 80 degrees centigrade, pwede na maglaga ng itlog dyan. Pero buhay sila. And then, the pH of 2 to 4. Kaya, may mga microorganisms na nagsusurvive sa tiyan natin, sa tummy natin, kahit na ang pH ng ating stomach ay around 2 ang value. Examples of this are the sulfolobus in hot springs and oxidizing sulfur bacteria. Yan. So, apart from Archaea Bacteria, ano pa ba ang mga members ng bacterial group? We have the Photosynthetic Cyanobacteria. This is formerly known as the blue-green algae. Kaya lang, di pa nakakalito, tinawag mong blue-green algae, pero hindi pala siya algae, bacteria pala siya. So, para hindi malito, palitan na lang. Use the cyanobacteria. So, this is present in both fresh and marine waters, and they can do photosynthesis. So, photosynthetic sila, just what you can see here in our photo. They are with chlorophyll. Pero ang chlorophyll nila not found in the chloroplast but only in lamellae. So, filamentous cyanobacteria, kaya parang ang hahaba niya tignan, no? Because they are filamentous. They produce heterocyst. So, they are also very good in converting nitrogen to ammonia. In other words, they are nitrogen-fixing bacteria, the cyanobacteria. And then, we also have the Eubacteria. These are all the modern prokaryotes. All modern prokaryotes belong to this group of Eubacteria. And we classify them, we identify them using their differences in the 16 subunit ribosomal RNA, ribonucleic acid. So, yan yung pinagkaiba nila, no? They have distinct morphological and physiological features. So, some of them are phototrophic, some are gliding, sheath, budding, appendage. Ibig sabihin ng appendage, parang may mga extensions pa siya, no? And they usually come in different shapes. So, there are 4 na parang major shapes ng bacteria. These are the cocci, or ang singular nito ay coccus. Cocci, other people would pronounce this cocci, it doesn't matter, no? That's the spherical. No, here you can see you have the single coccus, diplococcus, that means like 2 cells joined together, tetrads, 4, streptococcus, they are like forming chain, no? Para siyang kadena ng bilog-bilog na cells ng bacteria na magkakadugtong. And of course, the Staphylococcus. One best example nitong Staphylococcus ay yung tinatawag natin na Staphylococcus aureus. Staphylococcus aureus. Yun siya class yung nagkukos ng pigsa, nagkukos ng pimples. Yun yung bacteria na yun. Staphylococcus aureus. Yan. And then, we also have the bacillus or bacilli. No, that's the rod shape. So, meron tayong single bacillus, meron medyo fusiform. Ito siya class yung medyo parang matulis-tulis ang dulo. Some of this exist as, anong tawag natin? As an aerodynamic, ano ba tama? Para siya parang para sa aerodynamism. It's adaptation nitong bacteria kasi some of them are actually suspended in the atmosphere. And then the Diplococcus, Streptobacillus. Basta makarinig kayo ng word na strepto, that means it's forming chain. And there is the Coccobacillus. So there are like 2 cocci cells, but not really. It's just a bacillus that looks like 2 cocci cells. Okay. And then the helical or the curve, or what we call the spirillous and the spiral. Kasama na rin dyan class yung mga pleomorphic with no distinct shape. Minsan malapad, minsan mahaba, kulot-kulot, etc. So included in the helical and the curve are the vibrio or the comma, the spirillum, and the spirochete. Okay, let me just address on something. Somebody chatted from Aryan, malabo po ma'am sa akin, hindi ko masyadong makita. Alin ang hindi masyadong makita? Yung aking slides. Hindi mo masyadong makita. Bakit kaya? Sa iba ba class, ganun din? Malabo din ba sa inyo? Ma'am, sa akin din po, taasan niyo po quality ng video. Ganun, malabo sa inyo. Wait a minute class ha, tingnan natin. Oo, pala dapat kanina pa lang sa umpisa nagtanong na ako, no? Wait a minute. Paano kaya ito? Kasi naka-live na tayo. Wait ha. Sorry talaga. Sila po ma'am, magtaas ng video quality sa settings po. Pero ikaw, Paul, can you see me okay? Pero kayo Paul at saka si Densreal, nakikita nyo ba ako ng maayos? Ay, nako. Sa akin, okay po sa akin. Sabi ni Densreal, okay naman daw po sa kanya. Sa iba po. Pero mabili ko dito. Wait lang class. Let me try and figure it out dito rin sa settings ko, no? Baka meron akong pwedeng magawa. Kaso kasi dahil naka-live stream na tayo, baka pag mag-edit ako ay mawala tayo bigla. Sabi ni Shai May, okay na din daw sa kanya. Nag-adjust lang siya ng quality. Hindi ko ma-change sa akin class eh. Baka kasi biglang mag-end stream tayo. Sorry. Parang okay naman siya. Teka lang. Sabi nila, pwede daw kayo ang mag-adjust ng sabi ni Ma'am GD, si Ma'am GD ay nasa audience din. Sabi niya okay naman din daw sa kanya. Sabi ni Carla Jane, okay na rin daw sa kanya. Try nyo ng class, mag-adjust ng settings. Mag-try kayo ng 720 or 1080p. Kung hindi naman masyadong problema sa data, kasi usually yung mga YouTube naman ay free. Pwede naman ng 720, no? Sorry. Hindi ko ma-figure out dito sa aking settings. Sige, so I will continue, okay? Sorry doon sa, okay, sa kay Esther, okay na din daw. Sabi ni Ma'am GD, nasa group chat. So, change na lang daw kayo ng quality. Yan. Thank you sa mga nag-help. So, let me continue. Next na tayo. Okay, so now let's go to bacterial cell parts and function. So, ito yung representative bacterial cell natin. And these are all the different parts. So, we have the plasmid, we have the inclusions, nucleoid, ribosome, cytoplasm, the fimbriae, plasma membrane, cell wall, capsule, the pilus, flagellum. Okay, so isa-isahin natin ang mga parts na ito. Let's start with the cell wall. So what does the cell wall do? For bacteria, cell wall is composed of peptidoglycan. Peptidoglycan is a protein sugar molecule making the cell wall rigid. So pag basta cell wall, ang pinaka-function niyan lagi would be the shape, protection, diba? So basta cell wall. And then sa bacteria, it's very important ang cell wall kasi this determines the two physiological groups of the bacteria, gram-positive and the gram-negative, which is very important in classifying bacteria. So for example, you are given a task to identify or to classify, to identify an unknown bacterial cell or unknown bacterial sample. So, gagawin mo, pinakauna, you conduct the gram staining kasi it's very important to know whether it's gram-positive or gram-negative. So, ano ba ang characteristics ng gram-positive? Pag gram-positive ang cell wall, makapal ang kanyang peptidoglycan. Halos 90%, no, na merong peptidoglycan siya. Pag gram-negative, mas manipis yung kanyang cell wall. Okay? So, basta yun lang tatandaan ninyo. Marami ang mga pathogenic microorganisms in both groups, pero parang mas marami ang gram-negative bacteria. And, kapag ka nag-gram staining kayo, makikita nyo na under the microscope, you will see bacterial cell purple kung gram-positive siya. Purple, bluish ang color. Pero kapag gram-negative siya, makikita mo na reddish to pinkish ang kanyang color pag tinignan mo siya sa microscope. If you follow the protocol of gram staining. So, mamaya, ipapakita ko sa inyo kung paano ginagawa ang gram staining. No, meron tayong part ng techniques in the study of bacteria. So, madadaanan natin mamaya. Now, next is capsule. Capsule class is what we call the major virulence factor ng disease-causing bacteria. So, usually makikita mo ito siya kapag pathogenic ang microorganisms. Ayan. Now, capsule is also made up of polysaccharide and it keeps the bacterium from drying out. So, kung titignan natin yung figure, makikita mo na yung capsule ay nandito. So, next siya sa layer ng cell wall, na parang ang magkakasunod-sunod na wall ay ganito. Plasma membrane or cytoplasmic membrane. Sunod niyan yung cell wall bago yung capsule. Capsule yung pinakalabas niya. No? So, next is cytoplasm. Cytoplasm is where growth, metabolism, and replication are carried out. So, nandito yung mga structures na makikita mo dyan. And then, nandyan din yung enzymes, the nutrients, waste, and gases. So, if we go back to the figure again, here is the cytoplasm, this bluish structure in here. So you will see that all other cell organelles are suspended in the cytoplasm. And then we go to the cytoplasmic membrane or the plasma membrane. So in this photo, the plasma membrane is here, or sometimes this is called the cytoplasmic membrane. This is a fragile part that is located inside the rigid cell wall. In aerobic microorganisms, this one transports electrons and protons released during oxidation and converts the energy liberated into chemical energy usable by the cells. Kaya kailangan malaki ang kanyang surface area because it functions in a lot of things including nandoon yung nag-contain siya ng enzyme for synthesis, transport ng peptidoglycan, at iba pa, no? Nagsisecrete din siya ng extracellular hydrolytic enzyme, etc. So, pag sinabi ko, kaya importante na malaki ang kanyang surface area. So, how is the surface area increased in the cytoplasmic or the plasma membrane? So, that is achieved through the development of the mesosome. Yung mesosome is the invagination of the cytoplasmic membrane. So, you can see that here in our picture. This yellow here is our plasma membrane. And it produced mesosome as an invagination to increase the surface area for all the functions of the plasma membrane. So, please remember that. Next, we have the flagella. The flagella is the long and slender structure of the cell, which is generally providing the locomotion to many organisms, including the bacteria. So, it originated from the protoplasmic membrane. So, kung titignan mo dito sa photo natin na naipakita ko na kanina, the flagellum is here. In most instances, flagellum class is several times longer than the cell itself. So, kung ang cell mo ganito lang kahaba, yung flagellum can be a lot longer, several times longer than the cell itself. So, that is the flagellum. Fimbriae and pili. So, fimbriae and pili or pilos in singular. Pag fimbriae class, ang fimbriae at ang pili ay mga projections yan sila or the extensions ng cells. So, ano-anong function nila? Usually, ang fimbriae is para sa attachment. Shorter yan siya, straighter, pero yan siya ay ginagamit na pang-attach kapag yung bacterial cell ay mag-need siya to attach to a particular surface. It is the function of the fimbriae. Pero yung pili naman or pilos, that's usually functioning in the transfer of genetic material from one cell to another. Kaya ang tawag natin dyan ay sex pilos. So, ginagamit niya siya during conjugation. One cell transfers another DNA to a recipient cell. Kunyari, that's a DNA for antibiotic resistance. So, itong bacterial cell na to na hindi resistant sa DNA dati, binigyan siya ng cell na ito na resistant to antibiotic through the sex pilos, no, conjugation ng process, magiging antibiotic resistant na rin itong cell na to. So, that's the function of the pili. So, fimbriae and pili. So, fimbriae, you can see, itong mga medyo maiiksi na projections na to. These are the fimbriae. And look, the sex pilos here or the pilos here is longer than the fimbriae that is used to attach to another cell for conjugation. So, sabi natin kanina, the flagella is the locomotory organ of the bacterial cell if they are flagellated, but not all cells are flagellated. So, when they are flagellated, you can expect that they are motile, means capable of motion. So, if you look at this figure here, you will see an example of a bacterial movement. No, that's a bacterial movement aided by flagellum. Yan. Okay? That's a video I captured from the journey to microcosm site. Okay, next. This is the cell here showing a lot of ribosomes suspended in the cytoplasm. Unlike the eukaryotic cell, na napag-aralan nyo na yan dati sa cell wall pa lang, that there are differences between prokaryotic and eukaryotic cells. So, bacterial cell is prokaryotic cell. Wala siyang membrane-bound organelles. So, wala siyang tinatawag na endoplasmic reticulum, such as the rough endoplasmic reticulum, kung saan naka-attach yung ribosome. So, you will see here that the ribosomes are also suspended in the cytoplasm. So, ano ang function ng ribosomes? Hindi nagbabago, class. Marinig mo lang ang ribosomes, protein synthesis na yan kaagad. Okay? Hindi na natin masyadong i-detalye. Siyempre, marami pa yan, no? Kasi, pag sabi natin na ribosomes, marami pa. May tinatawag pa tayong mga ribosomal RNA, na nagpa-participate during protein synthesis pa rin, o diba? Basta protein synthesis. Anyway, in this microbiology class, we will have a separate lecture on protein synthesis only. Okay, next, nucleoid. Since, sabi ko nga, there is no membrane-bound organelle in prokaryotic cells, so instead of having a nucleus, meron siyang nucleoid. Nakikita nyo to? These are the genetic material of a bacterial cell suspended in the cytoplasm. Hindi siya nakabound in a nuclear membrane. So, freely suspended in the cytoplasm. That is what we call nucleoid. Pero nandyan pa rin ang kanyang chromosomal DNA. And, this still controls the functioning of the cell by controlling the protein synthesis through gene expression. So, next, the plasmids. So, plasmid, just like for example what is shown in here, are relatively small circular pieces of double-stranded DNA. Separate siya doon sa DNA na nandoon sa nucleoid. Parang extra DNA, extra genetic material siya ng bacteria. And that plasmid is also capable of self-replication. So, that one, no? Dito, oh. Next. So, how do bacteria divide? Paano ba nagdi-divide ang bacteria? Paano sila dumadami? Na bakit when you put your food on the table, kumain ka ng breakfast, tapos may natira, hindi mo naubos, nilagyan mo lang sa lamesa, tinakpan mo, nabalikan mo siya, hapon na. Pagdalik mo, panis na siya. Why? That means that bacteria and other microorganisms in there increased in number. But that's very rapid. Ang bilis naman, ilang oras lang, diba? Because some of the microorganisms can divide very, very fast in just a matter of few minutes. For example, 15 to 16 minutes by yung E. coli, dadami na yung isang cell, magiging dalawa. Yung dalawa, magiging apat, and so on and so forth. So, paano ba sila nagdi-divide? So, remember this. They divide through binary fission. Binary fission. Ibig sabihin, yung isang cell magiging dalawa, yung dalawa magiging apat, yung apat magiging 8, and so on. So, paano nangyayari ang binary fission? So, here is a cell, no? That cell has a copy of DNA. And that DNA will double, no? Magdo-double siya. So, mayroon ka ng original, mayroon ka ng copy. Para saan? Para it is a preparation for separation. So, paano siya magse-separate? Magkakaroon ng, kung ito yung cell, magpo-form siya ng invagination doon sa cell wall niya, and until eventually they will separate into two cells. So, binary, magiging dalawa siya. That is how bacteria divide, no? So, ulitin ko lang, the bacterial cell divide through binary fission. So, you have here a genetic material. That genetic material will double. So, you will have the original and the DNA copy. And then, that is in preparation for the pinching of the cell wall to start the division. Para this one cell will eventually become two daughter cells. More or less, an exact copy of the parent cell. Yan. So, binary fission. Next, let's go to the bacterial growth curve. Paano ba dumadami ang microorganisms? So, ito, ang bacterial growth curve na ito, class. Imagine mo to na naglagay ka, mayroon kang test tube. Dito sa test tube na to, sterile siya. In-sterilize mo. So, walang cell, wala kahit isa. Ngayon, naglagay ka ng 5 bacterial cell doon sa test tube na yun. So, 5 bacterial cell. Ang pinakauna, itong 5 bacterial cell na to, mag-increase sila in size. Hindi pa sila magdi-divide, mag-i-increase in size. No change in number, only change in mass. So they will increase in size. They will adapt to the new environment. So looking at how they will survive in that particular environment, titingnan nila ano bang pagkain nila doon, etc. No? Ngayon, when they are well adapted, oh, by the way, that part is what we call the lag phase. Lag, L-A-G, lag phase. In that time, there is no increase in number. So, paglagay mo ng 5 cell, 5 lang yan doon during lag phase. Now, next is, when the machinery starts running, these 5 will undergo binary fission. They will increase in number. So, yung 5 magiging ilan? 10. Yung 10 magiging 20. And 20 magiging 40, and so on and so forth. So, there is an exponential increase in the number of the cell. So, 5 lang yung inumpisa mo, magiging maraming-marami na siya, depending on the length of the generation time. Ano naman yung generation time? Take note of the generation time. Generation time is the length of time by which one cell will become 2. Na mag-undergo siya ng binary fission. That is what we call generation time. For other bacteria, 10 minutes, 15 minutes, 20 minutes, 30 minutes. For others, it can be few hours. But they can really grow very, very fast. So that is now what we call the log phase. Tandaan nyo lang yung logarithmic, no? Log phase. They will increase exponentially. Next is the stationary phase. Ito naman siya, no, pag galing dito sa log phase, dito sa papunta siya, bilis tapos magpa-plateau yung number niya. Hindi na magi-increase. Ibig sabihin nun, the cell stops dividing. Pero hindi pa namamatay. Maybe, nasaturate na nila yung food, nag-stabilize ang number. Pwede rin siya class na ang another option kung bakit nag-stabilize yung number is because the number of death and the number of cell division is equal. Kaya hindi nagbabago yung number niya. So, it's still stationary phase. Ngayon, at the point by which the toxic materials already accumulated, food has declined, and there is no more increase in resources, competition is very, very high, some cells will start to die. That is what we call the decline or the cell death. Magdaday na yung So, kaya pag nagko-culture kayo ng microorganisms in one culture medium, hindi pwedeng forever na yon, nilalagay mo siya ng forever na. So, you have to do sub-culture. Magsa-sub-culture ka, maglilipat ka sa bagong culture medium para may bago silang pagkain. Hindi mamamatay ang iyong culture na mini-maintain. So, that is bacterial growth curve. Next. Let's go now into techniques for studying bacteria. So, first is, we study bacteria for different purposes. One, we would like to know if the bacteria are motile or not, when they are moving or not. Or also, we would like to classify them or identify them. Kung what particular genus, what particular species. And so, we would like to study them microscopically. So, here we are. No, inevitable to use the microscope. Microscope is very important in the study of microorganisms because microorganism nga hindi siya nakikita ng ating naked eye. So, one satisfactory means of observing living bacteria, primarily to observe microbial motility, is what we call the hanging drop technique. Here in this figure, you will see that this one is a depression slide. O, yung depression slide, hindi yan siya slide na depressed, na nalulungkot whatsoever. It's a kind of glass slide na mayroong parang uka, meron siyang groove sa gitna. No, meron siyang ganun, oh. So, hindi siya flat na klase, but meron siyang, may depression siya. That's what we call. And then, next, what you need to do is get a cover slip. In the cover slip, you put petroleum jelly on the edges, sa edge nung parang square na cover slip. Tapos sa gitna nun, doon may ilalagay ang iyong drop ng microbial culture na titingnan mo under microscope. Tapos ididikit mo siya sa iyong depression slide. Pagkatapos invert. Kaya makikita mo, kaya bakit siya hanging drop? Kasi nakikita mo yung drop mo dito is hanging. And then you look at this under microscope. So dito yung microscope, no? Dito sa area na to. On top of this cover slip. So, ano ang makikita mo under the microscope kung magtitingin ka ng covers ng hanging drop na preparation? So, you will see bacterial movement, protozoal movement, kasi kagaya ng mga paramecium, ng amoeba, mabilis yan sila mag-move. So, kung ilalagay mo sila sa drop, contained sila, hindi sila tatakbo sa buong cover slip mo na mahihirapan kang habulin para lang ma-observe ang kanilang motility. Kagaya ng example na to. So, this is an example of what you see in a hanging drop preparation. Yan. No? So, nakakatuwa. Sana lang, no? Kasi wala tayong face-to-face. It could have been fun if you could do this in the laboratory. You prepare your own smear, you prepare your hanging drop, and then observe it under the microscope. But, sige, hanggang lecture lang muna tayo. And then, next, another technique to study is the use of stains. So, staining increases the understanding of microbial study, bacterial study. It helps you see the shape, it helps you see its staining reaction, it helps you see some very small parts, especially if you are looking at an advanced microscope. Kunyari, electron microscope. Ganun. So, ang simple staining, kaya simple staining siya kasi ang dali-dali niya lang gawin. No, kagaya dito sa procedure natin. So, meron kang smear dito. Ito yung bacterial culture mo na pinadry. Nilagyan mo ng stain, you just flood it. And then, you let it stay there according to the manufacturer standard. 5 minutes, 10 minutes. Tapos, pag na-stain na yan, i-wash mo lang, i-dry mo, i-observe mo na siya agad under microscope. So, ganun kadali. Kaya simple staining is just basically flooding your specimen with a stain and then drying it and observe it under the microscope. That's simple staining. Next, we have the gram staining. So, ito na yung sinasabi ko kanina that I will discuss with you the procedure on how to do the gram staining. So, gram staining was actually proposed by the Danish physician Christian Gram, where he wants to separate bacteria into two physiologic groups. That's the gram positive and the gram negative. So, how is it done? Here, class, and everyone viewing, you will need crystal violet, you will need grams iodine, you will need 95% alcohol wash, and the counter stain, which is the safranin O. Now, if you have the gram positive here and the gram negative bacterial cell, when you put the first step, which is the crystal violet stain for 5 minutes, when you see them under microscope, but you don't usually do that, but if you see them under the microscope, they will look violet or purple. Kasi crystal violet siya eh. Tapos next is the iodine. You put the grams iodine. What is the iodine for? Iodine is called a mordant. M-O-R-D-A-N-T. Mordant. It means that it enhances the absorption of the first stain you applied, which is the crystal violet, so that it will become deep purple, blue-purple for both the gram positive and the gram negative bacterial cell. Okay, so that's the iodine. And then next, you will have to wash it with the 95% alcohol. Kung natatandaan ninyo kanina, I said that the gram-positive bacteria has a thicker peptidoglycan, 90% peptidoglycan. So, thicker ang kanyang cell wall. So, kahit hugasan mo siya ng alcohol, hindi natanggal ang purple na na-absorb niya. Kasi makapal ang peptidoglycan. It will remain purple. For the case of the gram-negative bacteria, it will lose its stain. Magiging colorless siya. So, yun ang pinagkaiba nila, no, during the step 3 wash. So that, paglagay mo ng step 4 na wash, yung kulayan mo man siya, ang safranin kasi na stain is orangey, pinkish orangey, parang ganun ang color niya. So, itong gram-positive bacteria na purple na siya, kahit ano pang orange ang ilagay mo, anong kulay pa yan, it will not be colored orange or any other color but will remain purple. Kasi hindi niya na-lose ang color niya during the alcohol wash. But, the gram-negative bacteria, because it lost the stain during the alcohol wash, it will get the color of the counter stain. Yung last na stain na nilagay mo, ang ipoproject niya sa'yo na color kapag tiningnan mo under microscope. Ayan. So, you have the orange-red gram-negative bacterial cell in here. So, that's the difference between the gram staining. Next, the acid-fast staining. Itong acid-fast staining na to, class, para mabilis lang tayo. This is very good in identifying the causative agent of tuberculosis, Mycobacterium tuberculosis. Ito yung ginagawa nila, class, sa sputum test. Yung kapag gustong malaman yung mga may TB sa sputum test, yun yung ginagawa, acid-fast staining. And there are other staining techniques. Negative staining for capsules, endospore staining, Flagella staining, cell wall, and nucleic acid staining. Okay, so those are the other stains. Now let's go into preparation of pure culture. Okay. Here is an example of a Petri dish. Ito siya, class, gawa ito ng aking mga estudyante before pandemic. No, we grow microorganisms on nutrient agar. So, ito yung kanilang output. This, mga bilog-bilog na to, at mga irregular shapes, we call them the colonies. These are bacterial colonies growing on a solid agar medium. So, sinadya ko itong ipakita kasi these are isolated colonies. Ito siya, class, isang bacterial group, isang species of bacteria. Ito, ganun din, ito, ganun din, ito, ganun din. So, isolated. Pwede kang mag-culture niyan into pure culture. Pwede mong damputin using an inoculating loop at ilagay siya sa isang sterile culture medium. So, ito yung ating pinaka-target in preparation of pure culture, to have an isolated colonies so you can easily pick a colony that you wanna grow. So, what are the different techniques in preparation to pure culture? There are so many techniques, but isa sa pinaka-importante na skill na dapat matutunan mo before you do the pure culture is yung tinatawag na inoculation. Aseptic inoculation of microbial cells from a culture to a sterile medium, as shown in this text. You also have this in your modules. So, we flame the loop para mamatay kung ano man mga cells ang meron dyan, so it becomes sterile. If it is your test tube, you also put that near the fire so that contamination will be prevented. And then, you get a sample from there, you put back the cover, and you transfer that into a Petri dish, for example, or another test tube. And then you replace the cover, you flame again the loop so that before you put that on a table, wala na siyang buhay na cells, or else you might get contaminated. So in the laboratory, it is a very strict observance of aseptic method. So, ito na yung mga techniques. First, yung tinatawag natin na straight plate method. So, yung straight plate method, ganito yan ang susundan mo. So, may culture ka, galing sa test tube, i-spread mo yan siya sa iyong Petri dish. May pattern ito siya, class. Itong pattern na nakikita nyo to, ang tawag natin dito ay quadrant straight. Parang doon muna sa taas, dito naman sa gilid, dito sa baba. Ito nakikita nyo to, oh. Para siyang square, quadrant. So, ma-expect mo na dito sa initial inoculum, makapal pa ang growth dito ng bacterial cell. Pag dito ka na sa pangalawang streaking, medyo ninipis na yan ang bacterial cell. Pangatlo, mas manipis. Dito sa pang-apat, may expect na natin dito ang isolated colonies na gusto natin. Ayan. So, yan yung tinatawag natin na straight plate method. Next is, ito yung tinatawag natin na dilution for plate method. So, dapat, kung ito yung original inoculum mo, ipapadilute mo yung cells. Paunti ng pakonti yung cells para pag pour mo sa agar plate, konti lang ang cells na mag-grow. So, for example, here, this one is the original culture. You get 1 ml, you transfer that to a 9 ml broth. 1 ml again to another 9 ml and so on and so forth. So hanggang sa meron ka ng 1:100,000 dilution. Para pagdating mo dito, makikita mo, isolated na yung colonies. Kasi kapag ka ganito, di ba, they are overlapping, you will not be able to pick a colony without touching other colonies. So dito medyo okay na rin ito. Pero much better for pure culture is talagang super konti lang ang cells and very much isolated. So that is what we call dilution for plate method. So nasa pangalan niya na actually yung process. So first you dilute by pouring from tube to tube and then you pour that to the plate. So you dilute and pour to the plate so that you will have isolated colonies. Another method is what we call the spread plate method. So, sa spread plate naman, you just put a very small amount of bacterial sample and then you spread that using a spreader. So, by doing that, you are also spreading the cell so that approximately you will have a scattered and not overlapping or isolated colonies. Pero class, ang pinakamagandang gawin dito sa spread plate method na to is i-dilute mo rin muna siya. Similar dito, 'di ba? Bago ka nag-pour, dinilute muna siya. So pinakamagandang gawin dito, mag-dilute ka muna dito sa test tube before you put a sample on the agar plate and then you spread using a spreader so that you will have a better isolated colonies. Next. When you are studying bacteria, then you will see that there are different morphology of the colonies. So, merong different margins, by different colors, by different elevation, by different texture, and different shapes. So, this is one of the ways by which you can also identify bacterial sample. Maliban sa gram staining, you can also culture them on the plate and start giving the characteristics by putting the margin, putting the color, the elevation, texture, and shape. So, sayang nakalimutan kong maglagay dito ng sample ng plate. Pero balikan ko lang yung plate ko kanina. Yan. So, tingnan mo dito, may iba't ibang color, may iba't ibang morphology ng margin, may iba't ibang elevation. Kagaya lang nito, this one is, ang shape niya ay circular. Ang margin niya ay ganun din, circular. Tapos, kung titignan mo, ang color niya ay puti. Tapos, meron ding colors na yellowish, parang merong mga oranges. Tapos, ito merong lobate naman ang kanyang margin. Merong mga filamentous, etc. No? So, meron din tayong punctiform. Ito mga maliliit na parang tuldok-tuldok. We call that punctiform. So, these are examples of colonies characteristics na base dito sa ating table. No? Like rounded or punctiform. Yan. And then, if you see also some, kung para makita mo yung elevation, kailangan tignan mo yung iyong petri dish dito sa gilid. Para makita mo kung nakaumbok pa, flat ba yan siya sa nutrient agar, and so on. So, ito yung basis, no? You will do this pag gumawa na kayo ng culture ng inyong bacteria at home. So, do you have any question, class? You can type question sa ating question box. Naubos yata akong kagabi ng lamok dito sa pwesto ko ay. It's in a corner kasi. Ano, may tanong ba kayo? I'm waiting for your questions before tayo mag-proceed sa ating activity. Okay, so I hope na-solve niyo yung problema kanina na medyo malabo sa inyo, ha? So, any question, class? Kung wala, mag-type din kayo na wala. Kasi nag-iintay ako eh. Please let me know. Is it clear? Or is there anything that you would like me to repeat in the topics? We still have time. Hello kay Christine. Sabi niya, hindi ko daw siya student, pero gusto niya lang malaman. John Paul, wala po. From Jose, wala din po. Yung iba, do you have questions? Ang daming hindi naka-attend, ano? Parang hindi naman kayo masyadong dumami. Kasi ang nag-type ilan lang eh. Wala na rin from Charmy. Okay, so pag wala kayong mga tanong, let's go to our first activity. Our first activity, class, is a very short quiz, a 5-item quiz using Nearpod. Okay, here is the mechanics. This game is a game of pabilisan and patamaan ng sagot. Pag mali, nababawasan ang score. Pero kailangan mabilis din. Teka lang. Sige, Rafi, magsabalikan kita after kong i-explain. So, pagkatapos nung ating itatype ko sa sa chat box natin yung link, punta kayo doon, tapos ilo-load nyo yon, sundan nyo lang yung procedure, and then I will wait for you. Ipo-project ko yung progress ng ating lecture. So, before ko i-type yung yung link, may tanong si Rafi sa aking chat box. So, ano po ma'am ang magandang gamitin na gelatin powder po sa pag-culture ng bacteria? Okay lang po ba ang may flavor? Okay. I think that is pertaining to the activity that you have, no? Hindi. Hindi maganda ang may flavor kasi some of those may be harmful sa inyong microorganisms. Kahit na may flavor siya, ang totoo niyan, tutubuan pa rin siya ng bacteria. Kaya lang, pag may flavor, usually may kulay. Pag may kulay, mahihirapan kayong mag-distinguish kung may yellowish ba kayong bacteria. May kalimbawa, kung ang inyong bacteria ay, ang gelatin ninyo ay yellow, tapos meron pala kayong bacteria na yellowish, hindi nyo makaklasify na yellowish yun kasi yellow na yung color mo. So, yun yung problema kapag ka-flavored siya. And some of the flavors may be inhibitory to your bacteria. So, ang pinakamaganda is colorless, flavorless, tapos 100% coming from seaweed. Hindi siya synthetic na gelatin, but gelatin coming from seaweed para hindi siya matunaw ng inyong bacteria. Yan, Rafi. Yan ang sagot. Okay? So, are you ready for our activity? Okay, so I'm going to paste the link in our chat box. please click that and go to the page. Medyo mahirap lang kasi medyo loading yung iba. Pero antayin ko lang kayo. Antayin ko kayo na makapasok. I'm gonna join the game too. Pero syempre hindi naman ako makikipag-compete sa inyo. Pero bakit? Hindi na daw valid ang aking code. Bakit naman? BKX 54. Ay? BKX 54. Okay, nakapasok na ba kayo? Pangalanan nyo, ha? So, Halloween ang ating theme. Kaya naman magandang pang-Halloween. Yan. Okay. So, I have Jhonamie so far. John Paul. Asa na yung iba? We are waiting. Rafi is in. Okay, let's wait for others. Sana marami ang makapaglaro. Join everyone. Lahat kayo. Oh, by the way, may prizes tayo, ha? Para sa first, it will be 50. Sa second, it will be 30. And para sa third is 20. Okay? You can choose between load or GCash. Pero mas prefer kong ibigay sana na price ay load para sigurado ko na mapupunta talaga siya sa inyong pag-aaral ay magamit ninyo sa inyong mga classes. Where are the others? We are waiting. Come on. Loading ba? Okay. So we have Jhonamie, we have Shai, John Paul, we have Chevin, we have Rafi. Ang konti naman, 5. Nasaan na yung iba? Join the game kayo. May pa-premyo tayo. We have Kim Bernadette. Okay, 6 players so far. Nasaan na yung iba? Where are the others? Sila James, Mary Ann. Oh, si Anna Joy is in. Jose is also in. Esther Dawn. Densril, wala ka pa. Nasaan ka na? Si, Jhonamie, Aryan, Flordalisa, Ray Melvin, Jess, Karish, Gemma, Esther, Trisha, where na kayo? It's gonna be fun. Okay? How many are we? Let me see. There are 10 players connected. So there were only 9 plus me, that's 10. Shall I start na? Wala na bang gusto mag-join? Wala pa? Okay, I'm gonna start now. Yung iba mamaya, pagbigyan natin ng time, baka nag-loading pa ang kanilang mga internet. So for now, I'll wait for another one. Another one para 10 ang maka-join. Not including me, ha? 10. Isa pa. Hindi ka makapasok, Mary Ann. Just keep on trying. Just give it a try. Try and try. Isa pa, dali. I'm waiting for one more bago ako mag-start. Pag may nakapasok na isa, I will start. But the others, don't go, ha? Kasi, we will still have another chance later. No more na? Isa pa sana. Come on. Isa pa. Mahina ang internet. Nako. Oh, ayan. So, I'm gonna start the game now. Okay? Let's start in 3, 2, 1. Yay! So, first question. The oldest, structurally simplest, and most abundant life forms is called bacteria. Woohoo! Sino bang nauna? Ang bilis nilang umakyat, ha? Okay, bacteria with enzymes active only at high salinity are called thermophiles, acidophiles, halophiles, cytoplasms. The correct answer is halophiles. Next question. Formerly known as the blue-green algae. Ayan. So here, the correct answer is Cyanobacteria. Yay! You are leading. Next question. What bacterial shape is shown in the picture? Anong shape ba yan? Staphylococcus, Lactobacilli, Cytoplasm. What is it? The correct answer is Staphylococcus. Next question. Microscopic factories of protein found in all cells of bacteria. In other words, protein synthesis occurs in ribosomes, nucleus, Flagella. The answer is ribosomes. Okay? Who is winning? Wow! Jhonamie won the game. The second is Esther Dawn, and the third is Anna Joy. Ay, ang galing! Congratulations sa winner. So, Jhonamie, you got 50. Esther, you got 30. And Anna Joy, you got 20 from me. You can choose whether you want GCash or loan. You message me later. So, tingnan natin ang iba pa. Sino-sino pa ang mga nanalo. Wow. Sino-sino pa yung ibang nagka-points? Hi. Siguro itong iba na hindi nagka-points, masyadong mabagal ang internet nila. Kaya hindi sila nakakahabol na. Ay, Mary Ann, nakapasok ka. Kaso late na. So, pang-apat si Rafi. Pang-lima si Kim Bernadette. Pang-anim si Jose. Pang-7 si John Paul. 8 si Willie May. At 9 si Cherry. Okay. So, siguro internet ang problem. How was it, class? Kumusta naman? Ha? You want more? Ang galing ni Charmy. Nanalo. Okay, let me see. Comment back kung ano ang hindi ka makapasok. Okay. Ma'am, nag-e-error sa akin. Masaya, ma'am. Oy, masaya daw, sabi ni Esther. Kasi nanalo siya. Second. Okay, 30 ka sa akin, ha? Ang saya, ma'am. Haha. Pang ilang ka, Willie May? Ikaw ay pang ilan? Oh, pang number 8. Very good, very good, very good kayong lahat. Yung iba, ang problema lang talaga is mahina ang internet. Tapos na po ata, ma'am. Ma'am, isa pa po. Sige, Rafi, isa pa. Sana marami ang maka-join. Okay lang, Mary Ann. Let's have another go. Let's have another go. Okay lang, ha. Wait lang kayo. Okay, so here is our second game. Dapat marami ang makapasok, ha? Kasi pag hindi marami, hindi masaya. Dapat madami ang makapasok. Oh, lahat ng nasa audience, pati si Ma'am GD, sumali. Si Ma'am Aubrey, sali. Para mabilis, kung padami ng Kaya lang, bagalan nyo lang mag-join, ha? Para hindi kayo manalo. Natatawa ako, ma'am. Nape-pressure kami. Masaya yun yung pressure. Wait lang. Ayan na yung link. Sandali. Ako muna ay balik muna ako sa aking pag-join. Ay, ano ba yun? Nawala. Ano na yung link nun? bilis ni Mary Ann, nakapasok agad. Doon tayo sa Himalaya. Pili kayo ng character ninyo. Are you ready? Ayan, medyo marami-rami nakaka-join. Sige, wait tayo ng iba pa ha. Let's wait for them. What is it that you are viewing? Near 5. Come on, come on. More of this. More. More players, please. Where are the others? Okay, we have how many so far? Saan na yung iba? Come on. Come on. Yan, medyo marami-rami na. 13 connected. Let's wait for another 2. Sino mga nandito? Chevin, Kim. Dali ah. So we have Chevin, Kim, you have John Paul, Shai, Jose, Rafi, Charmy, Esther, Jhonamie, Mary Ann, Willie Mae, and who's the other one? Ay, dalawa. Bakit dalawa? Come on. Saan yung iba? I'm waiting. Wait lang muna tayo ng unti. Para makahabol yung iba. Mas marami, mas masaya eh. Mas maraming ka-compete. We have 13. More players, come on. Kayo na lang, wala nang iba. Ooh. Okay? Ready? Galingan nyo ha, para yung mga nanalo kanina, hindi na manalo, yung iba na naman ang manalo. Okay? We are starting in now. Where the chromosomal DNA of bacteria is located. Where ba yan? Located in ribosomes, capsule, nucleoid, pili. Correct answer is nucleoid. Let me see who's going to run very fast. Bernadette, number 5, Jhonamie, number 6, Karish, number 7, number 8, si Willie May, number 9, si John Paul, si Chevin, si Anna Joy, at saka si Shai. Siguro ano to, nagbagal lang kanila mga internet, kaya nagbaba yung mga scores. Okay, balik na tayo sa ano. Congratulations sa inyong lahat. Congrats kay Rafi. Okay. Sana na nga to. Hindi ko na-note. Teka lang, i-note ko muna yung mga nanalo para pwede nyo akong masingil. So, 1, si Rafi. Tapos 2, si Jhonamie. 3, si Mary. Kanina class, ang nanalo, number 1 si Jhonamie. Jhonamie ba? Number 2, sino na nga yung mga nanalo kanina? Sige lang, balikan ko lang mamaya. Okay. How is it? Kumusta naman ang laro? Nawalan na yung iba. Ang saya. Mas masaya yan kung alin as in sobrang madami. Kung naka-attend lang sana lahat, kung meron lang kayong mga around 50 students, tapos kahit ang makapasok ay sabihin na lang natin 30, kung madami lang talaga sanang naka-attend, it could have been more fun. Okay, so before I end, does anyone have any question about our topic today? Meron ba kayong tanong, class? Any question? Yun, nag-enjoy sila. Buti naman. Okay, we'll have another session of our online class, hopefully next week. So, another topic. If you are done with module 1, then you let me know so I can give the second module. Okay? Any question? None na daw. Ang saya po ng lesson. Okay, I'm very grateful and thankful that you were able to attend, and I'm happy that you enjoyed our session today, our class. I hope that more can attend next time. Please tell them. Inggitin nyo sila. Sabihin nyo marami kayo natutunan today. Please tell them that our class is fun so that next time they will also attend. I'll try to find a replacement for Nearpod that is more friendly to the internet connection kasi medyo talagang mahirap siyang i-load. Kung medyo mahina ang internet mo ay mahihirapan ka makapasok. So, mahirapan ka rin manalo kahit na alam mo ang sagot ka agad. Kasi pabilisan nga siya. So, kung sino mabilis ang internet connection, usually siya ang makakaunang makapasok ng sagot. So, thank you very much. That's alright. Kahit mabagal ang internet, we still had our good time today. So, that's all for this online class. I hope that you enjoyed. For those who missed it, they can still, including you can review this lesson through the YouTube channel. Thank you everyone. Bye-bye for now. I'll see you again next time. Bye! Thank you. Joyce, hindi po makapag-join. Hindi kaya ng internet connection. Sige lang, next time, you find a better and stable internet connection. Thank you. For those na nanalo, please don't forget to send me a message through the messenger. Yes, sana may ganyan, kayanin natin. Or try natin kung merong ibang platform na mas masaya or mas magaan sa internet. Thank you! Bye-bye! Bye everyone!